I'm sorry, it is the first Zoom conference for me. Okay, so first of all, I would like to thank the organizer for inviting me in this interesting congress. And good afternoon to everybody, or good morning, because in Italy is morning at the moment. So here my presentation entitled Recovery of Cellular Proliferation, Chromosomal Damage, and DNA integrity by metal sequester activity of cellulose-based nanomaterials, carried out in collaboration with chemists from Polytechnic in Milan. I'm a biologist at the University of Pisa, and in specific, today I present data recently published in Nanomaterials, mainly focus of the cellular level investigation to test the safety of a nanocandidate for water remediation process. Nanoremediation, as all of you well know, is referred to the application of nanotechnology to clean contaminated environmental matrices such as groundwater and wastewater. Selected materials need to be highly efficient in sequestering pollutants, completely reliable, and not exerting any toxicity for the biota. Many nanomaterials, as Alima this morning present in her presentation, like for example carbon nanotubes, nanozinc, nanotitanium, have been tested in remediation processes, but conflicting results have been reported concerning their potential ecotoxicity. So, for this reason, sustainable nanomaterials need to be safety for life environment and also could be developed from renewable sources, biodegradable and biocompatible. Also because selected nanomaterials for nanoremediation could remain in contact, exerting detoxification activity in environment for prolonged time. Among all the renewable sources, polysaccharides in general and cellulose in particular are emerging as starting materials. Here is presented the 6 principal steps starting from the cellulose nanofibers to synthesize our nanomaterial. For first, cellulose nanofibers are dispersed in water and sonicated, obtaining an homogeneous dispersion. Reagents are added to help the dispersion, and then the mixture was placed in molds and frozen. After that, water was removed by freeze and drying, and the reticulation was promoted by thermal treatment. And then the materials was washed in order to remove nanoreticulate components, and then the material was ready for our research purpose. Our aim was double: to test cellulose nanosponge metal sequester activity in environmental matrices, and in particular artificial seawater and fresh waters, and to evaluate cellulose-based nanosponge safety for organisms at the cellular level. For that, mollusk bivalves were used as model species and exposed in laboratory conditions. Mollusks are widely adopted both for ecotoxicology studies in marine and freshwater environment. Mytilus has been historically used as a bioindicator in marine pollution monitoring, and Dreissena polymorpha, or zebra mussel, was proposed as Mytilus' counterpart for freshwater study, due to their ability to accumulate heavy metals in soft tissues, including zinc and cadmium. Here, our experimental design was set up in Mytilus galloprovincialis gill cells for marine environment, being gill the interface between organisms and water. 5 experimental groups were prepared. One was the control, artificial seawater alone. One was the cellulose nanosponge alone, zinc alone, and eco-treatment, zinc and cellulose nanosponge in the same aquarium. Similarly, here the experimental design for fresh to test cellulose nanosponge activity in fresh water. The marine model, the freshwater, model was Dreissena polymorpha, and the cells are selected were hemocytes, being hemolymph the circulating tissue in zebra mussels. The same for experimental groups: a control, artificial freshwater alone, cellulose nanosponge alone, cadmium alone, and eco-treatment, cadmium and cellulose nanosponge. The exposure time was set up at 48 hours, both for zebra mussel in freshwater and Mytilus galloprovincialis in marine water. To detect DNA primary damage, comet assay was performed. It's a technique that in its alkaline version could detect single stain breaks, double stain breaks, and alkali labile site. Briefly, cellular suspension of gill cells or hemolymph directly was embedded in agarose and dropped in pre-coated slides. Then a Lisi solution was applied to degrade cellular membrane and nuclear envelope. DNA denaturation was possible in an alkaline buffer solution, and then electrophoresis was performed. Slides were stained with ethidium bromide, and the scoring was possible with a software. Detailed DNA is the parameter observed, and 2 slides for specimen and 10 specimen for experimental group, at least 50 cells per slide were scored. To detect chromosomal damage, cytomassay was set up. Cells were fixed in a prefixative and fixative solution, then dropped in microscope slides and staining with a Gemsa solution. The scoring was performed with an optical microscope to measure micronucleated cell frequency that could be arise from clastogenic or aneuploidogenic events, and to observe nuclear morphology abnormality frequencies. The principal nuclear morphology abnormalities observed in Dreissena polymorpha hemocytes were, as you can see here, nuclear bleb or nuclear bud, or the presence between 2 nucleus of a nucleoplasmatic bridge, the presence of vacuolated nucleus. To detect the rate of proliferation activity, the binucleated cells were observed. Moving to the results in terms of the absorbent capacity of cellulose-based nanosponge, shown a very good absorbent activity in artificial seawater. In fact, in zinc-polluted artificial seawater co-treated with nanomaterials, a significant reduction of more than 90% of zinc levels was observed. Similarly to what observed for cadmium dissolved in fresh water. For cadmium, the residual is nearly to the threshold imposed by European Union for drinking water. In terms of DNA primary damage, cadmium was found to be genotoxic at the selected dose tested. In fact, zebra mussels hemocytes exposed to cadmium show a loss of DNA integrity respect to bivalves exposed to the control. Moreover, cellulose nanosponge alone did not induce genotoxicity if tested alone. And interestingly, a recovery of DNA damage was observed in freshwater organisms co-treated with cadmium and sponge. Cadmium affects also the cell proliferation activity, as you can see, that's increasing respect to the control groups. Carbon nanosponge alone not affect cellular proliferation, and interestingly, in the co-treatment species, the effect induction of cadmium on cell proliferation activities was recovered. Both in terms of micronucleated cells and total nuclear abnormalities in zebra mussels hemocyte, cadmium exerts a toxicity. But when we observe cells exposed to cellulose nanosponge alone, the treatment not exert any toxicity, and in co-exposure, both in terms of micronuclei, then nuclear abnormalities, the levels are restored. Similarly to what found for the xenopolymorpha in freshwater, data from zinc exposed marine mussels show an induction of DNA primary damage restored by the co-exposure, by the sequester activity of cellulose nanosponge that alone did not exert tail DNA increasing. In terms of nuclear abnormalities in marine mussels gill cells, as you can see, zinc exerts a toxicity activity, while sponge alone did not exert any increase in nuclear abnormalities, and in the co-treatment group, the induction by the zinc was bring down the control level. Zinc was found to exert apoptotic induction while in marine mussels, gill cells exposed to CNS alone, apoptosis frequencies was comparable with the control level, and CNS absorption capacity brings apoptotic to the control levels in co-treatment specimens. So, in summary, both in laboratory condition for fresh water and seawater, the material, the cellulose, nanosponge material have demonstrated to be a good candidate in nanoremediation processes, being effective in the absorptions of zinc and cadmium and not exerting toxicity effects. In conclusion, cellulose nanosponges was found to be a safe and effective candidate in cadmium and zinc remediation process, being efficient in metal sequestering and restoring cellular damage exerted by cadmium and zinc exposure without altering cellular physiological activity both in gills, cells and hymocyte. Like future perspective, cellulose nanosponge could be increasing the synthesis process in order to reach, for what regard cadmium, the freshwater drinking thresholds. Also, this nanomaterial could be applied in nanoremediation simulation where pollutant mixtures are present and not only one contaminant, and thinking about projected field scale exposure. Are under analysis in this moment experiment similar to what presented here this morning, but where artificial seawater are contaminated with cadmium and artificial freshwater are contaminated with zinc. So I would like to thank everybody for their attention. So thanks from PISA and from all PISA nano groups. This year there's my email. Also, if you are interested in biological effect of yours nanomaterials, also for future project or investigation. Thank you.